So good evening everyone. We will be starting in a couple of minutes when everyone joins. Before we start, I would like everyone to go to the chat box and enter your full name and where exactly you are from and the institute where you are studying in at the moment. That will help a lot. I am audible, right? Hello. Yes. So Ujjwal is at the moment in Delhi, currently studying at Central University of Rajasthan. Oindrila is from Kolkata, either Kolkata. Nice. Nice to meet you all. So it is 4 o'clock. We will be starting at the moment. I hope my screen is visible. Yes, no, anything? Yes, okay. So I welcome everyone here to the week 3 a PMRF-NPTEL session on the course Introduction to Developmental Biology. The PMRF-NPTEL sessions are conducted in order to provide interaction-based learning between the students that are opting for NPTEL courses. So where Prime Minister Research Fellows such as myself can go through the assignment questions of previous years and help the student understand the concepts of the questions as well as the course material that was taught in this current week. So let me introduce myself. My name is Anuvab Chatterjee. I am a PMRF Fellow and PhD scholar under the supervision of Professor Jonaki Shen at the Brain Developmental Biology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. I will be the PMRF Fellow who will be conducting these sessions weekly throughout the course of the Introduction to Developmental Biology course, and we will be going through assignment problems and see how to solve them. So prior, if some of you have not joined or have not looked at previous videos, I will introduce briefly how these sessions go. First, we'll look at a particular question from an assignment of previous year and then try to solve the questions ourselves interactively. Then we'll go through a solution slide that will cover the concepts behind the particular question, and then we go through a concept slide that will cover the solution of the question and then answer the question itself. So if everything is clear and no one has any questions, we can proceed. Anyone wants to ask anything before we start? No, yes. A chat box message will do that. It is okay. It's fine, right? I guess it is. So we will start today. So this particular week, we went to developmental. Mohan Mukesh Malviya asked, "What is? Can you explain uh, what is anterior and posterior?" So when we consider, say, a three-dimensional object, like I can give an example of this duster right here. You can see the duster. Duster is visible. Yes, no. Here it is visible, right? So when we consider a three-dimensional object, we always know that it has a particular width, a particular depth, a particular height. This is how we define a three-dimensional object. But in the course of developmental biology and studying anatomy as such, we actually have to define these x, y, z, and zx axes ourselves. So these are named with respect to the medical terms or the anatomical terms as anterior posterior, dorsal and ventral, and the third axis is known as medial and distal. So how exactly should we explain this? So you take your hand, if you take your hand like this and just like, extend your arm, so your thumb is pointing to the top and your pinky finger is pointing to the floor. This up and down, like the thumb and the pinky finger are pointing in the anterior posterior axis. This is what the anterior posterior axis means. Along the thumb is the anterior portion of our body, posterior is along the pinky finger, like the legs and all that. Dorsal ventral are defined as ventral is the frontal portion here and dorsal is the back portion. And finally we have another axis. So this is more or less the height and the depth covered. So we have a third axis which is known as the medial and distal axis. Something that is medial is close to our body and something that is distal is away from our body. So my hand, when I extend it, it is distal from my body compared to my heart, say, which is medial. So these definitions are used in many aspects and most famously are used for most interestingly are used to define the positions of various areas in the brain. So you might have heard anterior medial hippocampus or dorsal neural invagination. Such terms are actually used in like many a times in biology in order to define a particular portion of a organ or our body itself. So Mohan, is that okay? I can actually even share my screen and show you an example. Yes, can you see this one? This let me open it. Yeah, can you see this picture? Yes, Mohan. So here you can see like right off the bat, there is a dorsal portion, that is there is a ventral portion and posterior anterior. These are essentially the defining features in order to define the position in a particular organ on with respect to the 3D space. I hope that is okay. Anyone has any questions regarding this? Everyone understood, right? What I've been trying to say. Yes, no, any chat messages would do. Yes. So this is an important topic that we will revisit when we learn the development of the embryo. There will be the zygote. When we say, look at it, it is essentially a single cell, but it develops. As it develops, it will there will be a symmetry breaking step that will define an anterior part of the zygote and a posterior part of the zygote that will initiate the development and morphogenesis processes. So this is something that we will revisit when we learn development of various embryos, probably when we look into the xenopus embryo and the Drosophila embryo as well. So with that covered, I would like to go back to the presentation. Anyone has any questions regarding this? I guess. So for the first question, this is the first question that we are going to tackle, and as I was saying, that in this particular week we have covered various aspects of developmental genetics and how various processes go on in our cells in order to regulate gene expression at various levels. The first questions will be regarding this only. So the first question is, for a particular mRNA, a northern hybridization experiment was carried out using total RNA isolated from cleavage stage and gastrulation stage embryos. Hybridization to intron specific probe indicated identical levels of mRNA in both the stages. However, an exon-specific probe indicated the levels of gastrula to be fourfold more than in cleavage stage. This is due to the four options are differential nuclear export of mRNA, differential stability of mRNA in the cytoplasm, selective inhibition of mRNA translation, and differential splicing of mRNA. So this is a box type of question, that is there are more than one correct. I would encourage everyone to try and answer the question in the chat box, and then we will proceed. So anyone trying to answer the question? Mohan says 4. Differential splicing of mRNA. So this is a more than one correct. So you can also try other questions, like other options. You can check if they seem correct or not. Mohan says differential splicing of mRNA. Anyone else that wants to try? Deepak says differential nuclear export of mRNA and differential stability of mRNA in the cytoplasm. Uchwal, Oindrila, anyone wants to give it a go? You have gone through the week's videos, I guess. Okay, so in order, let us move on and try to answer the question. First, let us know what exactly is northern hybridization experiment. Northern hybridization is an experimental technique that is used to detect the presence of a specific mRNA, or rather a specific mRNA against which you have a probe. So you know the sequence of the mRNA that you are trying to search, and using a northern hybridization experiment, you will be able to detect the presence of the probe. So what the question tells us is that when total RNA was isolated and a particular mRNA was probed using an intron-specific probe at two different stages of development, at the cleavage stage and the gastrulation stage, the intron-specific probe gave us identical levels of mRNA. But an exon-specific probe tells us that the levels of mRNA at the gastrula stage is fourfold more. What does this mean? So anyone can try and unmute themselves and try answering this. Like, what exactly is the difference between an intron-specific probe and an exon-specific probe? Like, this is a if you know the structure of the mRNA, you should be able to answer this. That what the intron-specific probe is detecting and what the exon-specific probe is detecting. Anyone wants to give it a go? Okay. So, an intron-specific probe will detect the nuclear RNA or the immature mRNA, and the exon-specific probe will detect the presence of a mature mRNA that will most likely be in the cytoplasm. So let us look into it. So this is an example of the question that we just saw, which shows us the ectoderm-specific gene expression on RNA processing of a particular gene known as Cy3A. So what they did here is they took particular cells from the ectoderm and other cells from the endoderm and mesoderm and tried to detect the presence of Cy3A intron-specific Cy3A mRNA using an intron-specific probe, and then Cy3A mRNA using an exon-specific probe. When they detected it, they found that the intron-specific probe in both the ectoderm and the mesoderm is the same, gives us the same level of mRNA. But in case of the exon-specific probe, the ectodermal cells give us a larger amount, while the exon-specific probe in case of endoderm and mesoderm gives us the same levels as the intron-specific probe. What this tells us is that in the exon-specific probe is able, what the exon-specific probe tells us is that the exon-specific probe tells us is that in the ectoderm the Cy3A mRNA is actually spliced and is in the functional form, That is, it is a mature mRNA that gets out into the cytoplasm. Whereas in the endoderm or the mesoderm, it stays inside the nucleus and is not processed. That is, functional Cy3A mRNA is only found in the ectoderm. So how can this be controlled? This can be controlled in two ways. First, they're preventing the exit of the mRNA or the mature mRNA from the nucleus of the endodermal cells, or the proactive degradation of the Cy3A mRNA where when it is present in the cytoplasm, so that it does not produce or produce any functional protein. Thus, to answer our particular question, there may be two ways in which this differential mRNA expression can be seen, which can be via differential nuclear export of mRNA. So in the gastrulation stage, the mRNA is allowed to be exported from the nucleus and then be translated, and by the differential stability of the mRNA in the cytoplasm. So it can be as such. In the cleavage stage, there are particular proteins that detect the particular mRNA and degrade it, so that there is no possibility of an exon specific probe detecting the presence of the mRNA. So these are the two answers. Differential splicing won't be a correct answer, because in case of differential splicing, both in case of there will be detection of both intron and exon specific probes in both the cases. So intron specific probe will detect the presence of a immature nuclear RNA, which will be detected even if there is a differential splicing. So both the levels will like the intron specific probe will be able to detect in both cases if there is a differential splicing. But if there is a needs to be a four-fold increase, that will only occur via differential nuclear export or differential stability of mRNA. Is that okay? Because this was a difficult question to think into, sink our teeth into. So anyone? So an exon specific probe will detect both mRNA and nuclear RNA, since exon is present in both. So what we can, since there is not a lot of information given in the question, we can understand that the exon specific probe might not be detecting just one exon. It might be detecting where the DNA sequence when two exons are actually together. That is, after the splicing process, when the exon 1 and 2 come together, the whole entire DNA sequence will be detected by the probe. If that is the case, then only the mature mRNA can be detected, because in the nuclear mRNA, the intron is in between and the probe won't be able to hybridize and detect it. Okay. Anyone else has anything to ask? Okay, so we move on to the next question. Which of the following are the examples of alternative splicing? The options are prolactin, alpha tropomycin, cordin, and casein. So this was something that was discussed in the lectures directly. The examples of alternative splicing, and this is also more than one correct answer type of question. So I would encourage everyone to drop in the options that they think is correct in the chat box. Oindrila says alpha tropomycin, cordon. Deepak says alpha tropomycin. Anyone else wants to try? Okay. Mohan says it is prolactin and alpha tropomycin. Okay. So we'll go through the examples themselves again that are given in the book. So the first example that we look into is the example of alpha tropomycin. So alpha tropomycin gene sequence is mRNA sequence is given right here. So we have a 5' UTR, different exons that are in colored patterns, and the gray bars indicate the introns, and then we have a general UTR at the end. So when during splicing, what alternative splicing does is it can during the process of splicing make sure that particular exons are incorporated, or particular parts of the introns are maybe also be incorporated depending on the type of splicing that the mRNA is undergoing, and this can in turn produce a variety of proteins. So, as we can see in the example of alpha tropomyosin, alternate RNA splicing forms a family of alpha tropomyosin proteins. So, the consecutive, yes, so this is what is written here. So, the first mRNA, that is the first mature RNA, actually does not have the general 3' UTR, so the splicing ends right here. Whereas in the second one, this striated 3' UTR is not there, and so on and so forth. In different parts, particular exons are incorporated, while in other mRNAs, the particular exons are not incorporated, and thus we have differential expression or the family of proteins we generated from the same mRNA. Is this okay? This was prime example of alternative RNA splicing. So this was our first example. The second is the alternative 3' splice site of cordon. So this is another example of, as I was saying, alternative splicing, in which instead of alternative exons being incorporated or not incorporated, alternative exons being incorporated or not incorporated, it might be the case that part of an exon is incorporated and part of it isn't. That will only be possible in what case? So if, say, the one of the splicing, one splice in one process of splicing, this the donor nucleotide is here and the acceptor is right here. So the splice sites remove this intron only, and the whole of exon number 9 is incorporated. But there can be a truncated variant in which part of the exon is also spliced out, and so we have a smaller part of the exon 9 incorporated. So can anyone tell me when can such a thing occur? When can an exon be partially incorporated or partially spliced out? What special or unique character should the exon have? So I would encourage everyone to try if any mutation occurs. That is correct, but this, in case of coding, this is naturally what occurs. Like a mutation is the mutation is kind of correct in case of mutation. There can be alternative splicing that are produced in it, and that causes the anatomy to go into a disease state. But there needs to be something here, like what is the mutation, what can occur that will allow incorporation, presence of similar sequences. Okay, so have we heard of, like, do we know how exactly splicing occurs? Does anyone here does not know how exactly splicing occurs? Okay, I assume then everyone knows that there is a splice donor site and a splice acceptor sites. These are particular DNA sequences that are recognized by the spliceosome, the protein complex that is what controls the splicing of a particular mRNA. And depending on the presence of a splice acceptor site, the intron is recognized. So there is an acceptor site right here, which is recognized by the spliceosome, and thus the cut is present here. So what exactly is present in this coding mRNA is there is a splice acceptor site present in the middle of the exon as well. So the spliceosome can, instead of recognizing this site, can recognize this one as well, and thus then produce a truncated variant by splicing out entire intron part of the exon 9, as it still thinks that this is the entire thing is an intron. Is that okay? Are we? Everyone understood, right? Okay. Yes. Okay. So I would really encourage everyone to try and interact, because these sessions are built in order for classroom based interaction to occur, just that we are on a Zoom. Yes, Deepak asked if I can repeat. So during the splicing process, if we go back to our molecular biology classes, there are particular sequences that are present. So there are particular sequences that are present on the starting of the intron and particular sequences present at the ending of the intron. These are known as the acceptor and the donor sites, which are actually recognized, and the cut is then made by the spliceosome. In case of normal mRNAs, there is an intron-specific donor site right here and acceptor site right here, and thus the intron is spliced up. But in codon mRNA, there must be a acceptor site right here in the middle of the exon. So when the spliceosome sits and tries to recognize the acceptor site, instead of this intron acceptor site, it can also recognize the exon acceptor site. And the spliceosome then considers the entirety of the intron and partial exon 9 as the intron that needs to be spliced out. And then it splices out and ligates this acceptor and donor sites to form the truncated variant. Is that okay, Deepak? Yes. Okay. So after looking into it, which are the following are the examples of alternative splicing? We have alpha-tropomycin and codon. So prolactin and casein will just show up in a bit, and we'll look into how they are regulated. Can anyone try and venture what is the regular? How is prolactin and casein regulated? mRNA is regulated. Can anyone try to answer that? Because we will have a question momentarily on it. Stability, very nice. So we have someone who has actually gone through the lectures very well. Yes, the stability of the mRNA is the answer. Stability of casein mRNA is stabilized by the presence of prolactin, and thus it can stay in the cytoplasm and produce try and allow transmission of the casein protein for a longer time. So we move on and look at the next question. The question asks us, eukaryotic messenger RNAs are often found as circles resembling crest rings. The crest or the head part contains there are four options. First is E1F4E, splice acceptors, poly A binding protein, and E1F4G protein. So essentially, eukaryotic messenger RNAs are not like a long string as we have drawn them again and again from the 5' end to the 3' end. They essentially form a circular structure with some proteins binding at the ends, 5' and the 3' ends. So the question asks us, what exactly are these proteins? So this is also a box type question, so there are multiple options that are correct. So I would encourage everyone to try and answer this. Shian says B and C, splice acceptors and poly A binding protein. Anyone else wants to try? Oindra says it is 1 and 4, E1F4E and E1F4G. Mrilanini says it is 1 and 3, E1F4E and poly A binding protein. Okay. So anyone else that wants to go ahead and try? Mukesh says 1 and 4, E1F4E and E1F4G. Okay. So let us look at the solution slide and try and understand this. So these are the crest-like circular structure that I was talking about. The head structure that is glowing a lot under the atomic force microscopy is the other proteins that are present. So this is a translationally active protein, and I would say it's nice. This can shift and we can see it perfectly clearly. And here we can actually see that in a translationally active mRNA, there are various proteins that are involved. These include the 4E and 4G proteins. 4E binds to the 5' cap, and there is a poly A binding protein that binds to the poly A tail. This poly A tail is something that is introduced in the cytoplasm. So during the processing of mRNA, we know that the splicing and poly A tail addition occurs in the nucleus itself, but it can also occur in the cytoplasm. So in many mRNAs, the addition of a cytoplasmic poly A tail is what changes it from a translationally dormant state, where there is no poly A tail, to a translationally active state where a poly A polymerase adds a poly A tail to which a poly A binding protein binds, which then causes the interaction between 4G and 4E, and then a ribosome can come and bind. In case there was no poly A tail, there is no poly A binding protein, and instead the protein masculine binds to 4E and CPEB, another protein, and this does not allow the 40th ribosome to come and bind, and thus there is no translation. Thus, in a translationally active mRNA, there are proteins such as the 4E, 4G, and PABP, that is poly A binding protein, binding to the crest or the head of the mRNA. Thus, in answer to the particular question, we have three correct answers, which is E1F4E, poly A binding protein, and E1F4G. Splice acceptors are essentially proteins which are present inside the nucleus itself. So they won't be present in the crest-like structure when we see which are seen in the cytoplasm cells. So anyone has any questions on this? I guess so. We can move. So we come to the question right here. So how does that we were discussing just before? How does prolactin affect the expression of casein? The options are: it activates the nuclear processing of casein mRNA, it promotes nuclear export of casein mRNA, it inhibits the splicing of casein mRNA, and it promotes the stability of casein mRNA. So since we have already, doesn't PABP form the base of the ring or not crest? So base, essentially, if we look at this structure right here, the string-like structure itself, if I can zoom in, the string-like structure is the mRNA, and all the proteins are too large, and that is why it looks like the head of a structure. And if we go down here, you will be able to appreciate that this entire protein complex is what looks at the like the head of the protein. So there is essentially is no base of the ring, but like if you have worn a ring, you will know that the jewel on top of the ring, or the signet that is top of the ring, forms a broad structure, and this protein complex is what forms that entire structure. Okay, so these are the proteins that are binding to the mRNA, and the mRNA itself is the ring. Yes. So this one we have already, Oindrilla has also already answered, so I am not going to go through it again. The prolactin affects the stability of the casein protein. So this is the example that is given. So in the absence of prolactin, in a pulse chase experiment, so what exactly is a pulse chase experiment? In a pulse chase experiment, what we do is that we label a particular protein or particular cell or whatever with a label that stays with those particular cells or proteins. So how it is done is that rat mammary gland cells are given radioactive RNA precursors or a pulse, and after a given time were washed and given non-radioactive precursors. So those RNAs that are produced using the radioactive precursors will be radioactively labeled, and everything else that comes after that using the non-radioactive precursors will not be labeled. So essentially what it does is that at a specific time point you are able to detect or label every RNA that is present inside the cell, and after you remove the pulse, the cell goes back to normal non-radioactive precursors. So you have a particular population that you can track, and like how are these particular RNAs interacting, or what is the lifetime of these particular RNAs, is something that you can track over time, because no more radioactive RNAs are being produced. Only those population that you had labeled are the radioactive ones that are inside the cell. So in these experiments, in the pulse chase experiments, we can look into the half-life or the entire time period in which a particular protein is present inside the cell. These pulse chase experiments are also used to mark and detect the half-life of particular of cells. And very famously, the pulse chase experiments were used to detect the presence of Okazaki fragments, if you remember, going back to mol bio classes. So this is the experiment that was done, and in this procedure, the casein mRNA synthesized during the pulse time was labeled. And when a plot is given, we can see that the percentage of casein mRNA drops down dramatically when there is no prolactin present, but in the presence of prolactin, it stays for a longer time. So the half-life is 1.1 hours without prolactin, and in the presence of prolactin, it goes to 28.5 hours. So, but how does this help? The half-life of a mRNA help in the production of the protein. Can anyone try and guess this? So, like, say the mRNA is present for 28 hours instead of 1 hour. So, how does this affect the protein turnover? Like, it is staying in the cytoplasm. What extra thing is happening? Anyone tries to answer that? If it is too difficult to answer on chat, you can just unmute yourself and go ahead and answer. Okay. So no one has a clue why the half-life affects the protein turnover. I am audible, right? Hello? Yes. Okay. Okay. So, so let me go ahead and answer this. So, protein, possibly lactic stability is increased. No, see, we are talking about casein. Casein is the mRNA whose stability is increased. Prolactin is a hormone. So when the hormone is detected by the cell, it allows the casein mRNA to be stabilized, and thus this casein mRNA stays in the cytoplasm for a long time. So the question is, what does what changes? How is it effective in regulating the gene expression of casein? So what happens is that we have seen when a particular mRNA is translated, what happens? The ribosome binds to the 5' cap, translates the entire mRNA, and then we have a new polypeptide that is synthesized, and then that is then transported to the endoplasmic reticulum. But what happens to the mRNA? What happens is that after the ribosome is done, the mRNA is still present in the cytoplasm. Then another ribosome can come and bind and produce another peptide. So essentially, the mRNA can act as a template for multiple rounds of translation. It is not that once the mRNA is translated, it is gone. It entirely depends on how long the mRNA is translationally active and present in the cytoplasm. So, say our casein mRNA was present only for 1 hour. Thus, when prolactin is present, the casein mRNA can now be used as a template for 28 times or 28-fold increase in the peptide production, because it is still there in the cytoplasm. So same template can be used again and again and again. So in essentially, you give the protein turnover a huge boost. Is that did we get it? The casein mRNA essentially being in the cytoplasm allows it to be used as a template to produce new polypeptides again and again and again, and thus we are producing, thus the gene expression of the casein mRNA increases dramatically. It's okay, no. Did you understand or did you not? You can just yes. Okay, so we move on, and obviously, the answer to the question was it promotes the stability of the casein environment. So next, we move on to another interesting topic, which is that of microRNAs. The question is, how do microRNAs influence the expression of their targets? The four options are: they facilitate or inhibit nuclear export of target mRNAs, they block RNA polymerase from binding to the target promoter, they recruit proteins that block translational initiation, and they recruit proteins that cleave the nascent peptide. So this is also a square type of box answer, though that is we have more than one option correct. Deepak goes ahead and says that it is 2 and 4. That is, they block RNA polymerase from binding to the target promoter, and they recruit proteins that cleave the nascent peptide. Oindrilla says 2, 3, and 4. That is, in addition to 2 and 4, they recruit proteins that block translation initiation. Anyone else wants to try and answer this? Brinalini says 4, 2, and 3. That is, they recruit proteins that cleave the nascent peptide, they block RNA polymerase from binding to the target promoter, and they recruit proteins that block translation initiation. Sriyans also says they block RNA polymerase from binding to the target promoter, and they recruit proteins that cleave the nascent peptide. Mohan also follows up. He also says the same. RNA polymerase binding to the target promoter and proteins that cleave the nascent peptide. Okay, so let us look into the solution slide. MicroRNAs, what microRNAs essentially are short stretches of RNAs that have complementarity to the mRNA that is being produced. And what they do is they go and bind to the mRNA. The binding to the mRNA helps a particular complex known as the risk complex in recognizing the mRNA. If there is a lot of complementarity between the mRNA and the microRNA, then the risk complex can stop the translation or cleave the mRNA entirely. So the microRNA complex, including numerous proteins, bind to the microRNA and form the miRNP, that is the microRNA microRNA protein complex, and can block translation in several ways. These include blocking the binding of the mRNA to initiation factors or ribosomes. Am I audible? Because it is showing to me that my internet connection is gone. Can anyone hear me? Audible now. Okay. Okay. So what I was trying to tell you is that microRNA is complementary to the mRNA that it is trying to regulate. MicroRNA is incorporated into the risk complex, and when the risk complex recognizes a mRNA that is complementary to the microRNA, it can either stop the translational process or start cleaving the mRNA itself, not the nascent polypeptide. No, it can cleave the mRNA itself and also the nascent polypeptide. So there can be three ways in which the microRNA can initiate the process of translational regulation. The first one is the initiation of it can block the initiation complex, the translation initiation complex. It can allow the endonuclease digestion of the mRNA itself, so it will cleave the mRNA, or it can allow the degradation of the nascent polypeptide. That is, it is allowing the degradation of the peptide itself, so that no peptide is produced. But here is a question: So why are microRNAs? Why do you think microRNAs are required? Anyone, like, other than regulation of the gene products, can there be another different aspect towards microRNAs, like something else that the microRNAs can help in? Play a role in cell differentiation. That is correct. They play a role in cell differentiation by regulating the gene expression of various genes. So, in a particular gene cell precursors, maybe many of the genes were expressed, but in subsequent progenies or subsequent differentiated cells, these protein and gene expression is stopped by the microRNAs. But that is also gene expression that we are talking. There is another aspect towards microRNA, that is in case of that is they can yes, they can act as a defense mechanism towards viral infections. So what happens is that when a viral virus comes into a particular cell, it tries to produce its own viral proteins, that goes through the process of producing a viral RNA, which is then translated by the cytoplasmic components. What the microRNAs can do is that they recognize the viral RNAs themselves and start cleaving them. Like the entire microRNA regulation process occurs on the viral proteins, and thus no viral proteins are produced. So they can act as a defense mechanism. So how do microRNAs influence the expression of the target? It will be all the last three of them, 2, 3, and 4. They block the RNA polymerase from binding the target promoter is obviously not correct, because RNA polymerase is the protein that produces the RNA. The microRNAs influence the translational step, that is, they block the translational initiation and they recruit protein that cleave the nascent peptide. So it is actually 3. Is that okay? The discussion was clear, right? You understood what I was trying to say? Yes, no. Yes, okay. So so we have, for a long time we have been discussing how exactly gene expression is regulated, and this question probably concludes everything. The question asks us what the control of gene expression at the level of translation can occur by the following options are there: differential mRNA longevity, alternative RNA splicing, selective inhibition of mRNA translation, and ribosomal selectivity. So we have 4 options. The control of gene expression at the level of translation. 3 and 4. Selective inhibition of mRNA translation and ribosomal selectivity. Anyone else? Rinalini says alternative RNA splicing and selective inhibition of mRNA translation. Mohan says differential mRNA longevity and selective inhibition of mRNA translation. Anyone else wants to try? Shriyan says alternative RNA splicing and selective inhibition of mRNA translation. Deepak also says 2 and 3. Okay. So we look at going to the solution slide and look into it. So control of gene expression can occur through these 3 ways. The first is differential mRNA longevity and stability. We have already seen an example of this in the casein mRNA and prolactin that we just discussed. So this is one of the ways. Another way is that selective inhibition of mRNA translation, which occurs in cyclins and growth factors in oocytes. So selective inhibition means that particular genes are allowed to express only in particular regions of the oocytes, which, as I was talking about, produces the anterior posterior asymmetry. That is, one portion of the oocyte can be defined as the anterior region and another will be defined as a posterior region. This is actually produced by selective inhibition of particular mRNA translations. And the example is cyclin and growth factors in oocytes. And finally, it can also be via microRNAs based regulation of translation, which we have just discussed. And an example of it is Lin14 mRNA regulation by Lin4 microRNAs. Okay, all of this is already what we discussed, and thus the question is gets everything together. So the answers to the question are differential mRNA longevity and selective inhibition of mRNA translation. Alternative splicing is not there because there is a post-transcriptional modification, and ribosomal selectivity is, I do not think there is something as ribosomal selectivity. So this was just there. Okay. Okay. So the next question is, a mutation in the dash of the myostatin gene led to the formation of larger muscles. So this, let me give the context to this particular question. So there is a particular strain of mice known as mighty mouse, in which there is a huge lot of muscle production in comparison to normal wild type mice. This occurs by a particular mutation that occurs in the myostatin gene, which gives rise to larger muscles. So the question is, where exactly does the mutation occur? So this is something that we have already seen in the lecture. So yes, Mrinalini and Oindrila both say the mutation is in the splice site. Kavya also says it is in the splice site. Okay. So let us move on and look into it. It is still not visible. So here is the example of what exactly occurs. So in the wild type, myostat in the wild type myostatin gene, we have a normal splicing that occurs. There is exon 1, exon 2, and exon 1 and 2 are joined together after the nuclear RNA processing to form a functional mRNA and the functional protein. In the mutant mice, what occurs is that there is a particular GTA AGT sequence that is present in the intron. What happens is that with the mutation, what occurs is that a G gets converted to a A, and this essentially acts as the splice acceptor sites that I was talking about earlier. So what happens is that it acts as the splice donor site. So instead of exon once this end, the boundary between the orange and the gray region being recognized as the splice site, this is recognized as a splice site, and so this entire portion of the exon and partially the intron is incorporated into the mRNA. So we have a mRNA that looks like this. So part of the intron is also incorporated into the mRNA. What now occurs is that since there is a termination codon present in the intron itself, so when the ribosome starts translating, there is a premature end to the translation, and only exon 1 is translated, and which obviously gives rise to a completely non-functional protein. This now, so in the wild type we have a functional myostatin, and in the mutant we have a non-functional myostatin. What then occurs is that myostatin works in order to stop the muscle cells from dividing, and thus the growth of muscles stop at the particular time. But non-functional protein cannot obviously stop the muscle cells from dividing, and a lot more muscle cells are produced. Muscle hypertrophy is there, and the mighty mouse is generated essentially with a lot more muscles. So this is what exactly occurs in this. So anyone has any questions? So here is a dissection of the mice. So there is a dissection of the mice. So we have in this particular normal wild type mice, you can appreciate the change in muscle density between the both of them. So the answer is the mutation is present in the splice site. Okay, is that okay? Yes, no, maybe. Yes. So we move on and answer the question: which of the following is are not involved in mRNA transport? The options are myosin, actin, microtubules, and kinesin. So mRNA can be transported to specific areas. So if when imagining the eukaryotic cell, we oftentimes miss that there is essentially a railway or a roads and a railway present inside the cell in the form of the microtubules. So any organelle, such as a lysosome or a mitochondria, can attach to a microtubule and start walking along the microtubule to go to different parts of the cells. This is also true for protein. So if you ever thought, Jay, a particular protein, how is a protein that is necessary for a for mitochondrial membrane, does how does it reach to the mitochondria, or a cell receptor that is that was obviously a protein that was being transcribed in the cytoplasm, how does it know it has to reach the cell membrane, or what exactly takes it to the cell membrane? It is the microtubule network. So this also is true in case of mRNA. So organelles, proteins, mRNAs can attach to the microtubule network and then be moved to various parts of the cells where they are actually required. So the question here is, which of the following is involved in mRNA transport? The options are myosin, actin, microtubules, and kinesin. We have four options. So Mrinalini says myosin and actin. These are not involved. Yes, the question is not involved. Myosin and actin. Anyone else wants to try? Okay. So we here is the example of cytoskeleton transport. So dynein and kinesin and microtubules are help in actually the movement of mRNAs to various parts. So this is also another symmetry breaking step that I was talking about. Preferentially, some mRNA, such as the becoid mRNA, is taken to the anterior end or one of the ends, and the Oscar mRNA to the other end. This essentially is what the oocyte comes with. So the already in the Drosophila oocyte at least the symmetry is already broken by differential transport of mRNAs to specific parts. So this is something we'll look into as we look into Drosophila embryogenesis. How specific mRNAs and specific proteins such as becoid, caudal are present that help in distinguishing the anterior and posterior fates in Drosophila embryo. So the answer to that, which of the following are not involved in mRNA transport? It is myosin and actin. Microtubules, kinesin, and dynein are involved. So actin is a cytoskeleton protein which helps in maintaining the cytoskeleton of the cell. Mrinalini, can you try and answer what myosin is involved in? Do you know? Mrinalini, I assume you know something about myosin because you were correct about the question. So you know, do you know that what exactly is the function of myosin? Yes, Mohan is correct. It functions as in the movement of muscles. So if myosin in muscle contraction, yes, myosin in muscle contraction is correct. So this is a large topic, but myosin, what myosin does is helps. It is essentially the protein behind muscle contraction, which helps in muscle contraction and relaxation, depending on the presence of calcium ions. So that is a lot longer conversation, but it is correct. The myosin is involved in muscle contraction. Mohan, Kavya also is right. It aids in muscle movement. Exactly muscle contraction, but it is correct. So this is a numerical type. In a dihybrid cross between pure breeding pea plants bearing round and yellow seeds and wrinkled and green seeds, 100 plants bearing round seeds were obtained in F2 progeny. What percent of these 100 plants will have green seeds? Round off your answer to the whole number. So this is a numerical type. So we have moved on from gene regulation to genetics. So we have a pure breeding pea plant with round and yellow seed and wrinkled and green seed. And these were allowed to go to the F2 crossed and allowed to go to the F2 progeny. And out of all the progenies, 100 plants have green seeds and 100 plants have round seeds. So what percentage of these will have green seeds is the question. This is obviously a numerical type that we need to actually work out. So does anyone want to actually work this out now? Like, it will be a scribble or two on the paper. If you have a paper, you can go ahead and try. Or rather, we can just go to the solution slides. Is anyone is anyone interested in trying? Okay. No, I guess. So I have already solved it. So we can also solve it now if we want, if this does not suffice. So here is the dihybrid cross that we are talking about. We have a yellow, pure breeding yellow and round seed, and a pure breeding wrinkled and green seed. So obviously we know that the capital Y, this is obviously small y because it's allele. So there is no GG or wrinkled. For wrinkled we do not have a WW. We have YY and R because these are recessive alleles to these dominant genes, dominant alleles. So the gametes will have Y and R and small y and R, and obviously after the F2 cross we have a 9:3:3:1 ratio. So this is also here where I have worked it out. So at the F2 generation we have VL around 9. We have a ratio of 9:3:3:1 for all the phenotypes that were observed. So by the problem we know that round seeds we have 100. So what are the round ones? It could be a yellow round or a green round. So essentially we have a 12 out of 16 ratio of the total seeds, which gives us 100 round seeds. So this portion, so 9 plus 3, 12 out of the entire 16 is when we fraction of the total seeds is actually round. So essentially what we are trying to say is that a 12 out of 16 fraction of the total seeds is the round seed, which is 100. And thus the total seeds come about to just cross multiplying 100 into 16 by 12. Next what we do is the green seeds. So green seeds out of the total round seeds. So how many round seeds did we have? We had they wanted to ask green seeds out of the round seeds. So that is a green round seed, which is 3 out of 16 of the entire fraction. So the green round seeds equal 3 out of 16 of the total fraction, total number of seeds, which is this quantity that we have kept without calculating, so that everything cuts off, and we get the final answer as 25. Okay, so the answer to the question is 25, and this is how we did it. But obviously this was very if anyone has any questions, because obviously this might just go over your head because I went too fast. Is this okay that we have just used fractions? So you can also say like old school algebra, that let the total number of seeds be x, and so every one of them will be 9x, 3x, 3x and x. We add all of them total, and then this 9 plus 2, 12x equal to 100, and then calculate. Is this okay? Does anyone have any question? Kavya says it is okay. I assume everyone else is also fine with how we did it. So the answer is 21. So some these questions will be asked, very basic mathematics, but you have just to keep your head when answer these questions, because something like this, like what percent of the 100 plants will have green seeds? Many a times we might go about calculating the total amount of green seeds, that is then the fraction will be converted from 3 by 16 to actually 4 by 16 if you go wrong. So these things are something that we have to keep in mind, that they have asked out of the 100 plants the number of green seeds, that is they have essentially asked the green round seeds instead of the total green seeds. So we move on. The next question is, in Drosophila, if recombinant frequency between two genes is 10%, then these two genes are, four options are, very tightly linked to each other, located on the same chromosome, located on two different chromosomes, and independently assorted. Okay, so anyone wants to try and answer this? One and two. Very tightly linked to each other and present on the same chromosome. Actually, this is only one option correct, so most relevant option is something that we have to choose. Hmm? Did something happen? You did. Hmm. Is my screen visible? Hello? Is my screen visible? Am I visible? What is happening? Hello? Yes, yes, yes. Okay. Yeah. So that is the question. That is, if the recombinant frequency between two genes is 10%, these two genes are very tightly linked to each other, located on the same chromosome, located on two different chromosomes, and independently assorted. Okay, so anyone wants to try and answer this? Okay, Mr. Three. Okay, located on two different chromosomes uh, is what Mohan says. It is located on two different chromosomes. Kavya says it is uh, located on the same chromosome. Deepak said the first two. So let us look into it. This is a. Let us try this. Okay, for some reason my camera is not working. Okay, this is what the entire link genes concept is. So, in case of a F1 dihybrid in Drosophila, obviously we name it in this way, that a particular allele which is the particular dominant form of a particular allele is given as a plus, and the recessive is not given anything. So, in this particular fly, we have a black and vestigial winged fly, which has dominant alleles on the same chromosome and recessive on the other. During meiosis, crossing over can occur obviously, and this can give rise from two chromosomes, two varieties that should have been there, that should have independently assorted, can give rise to four different gametes, in which due to crossing over we have one gamete with both the recessive dominant, one with both recessive, that should have occurred both of these. But we also have recombinant gametes, in which one gene recessive and other gene is dominant, and we are mating it to a fly which is a double mutant, that is in both the chromosomes, all its alleles are recessive. When we cross these, we have these number of test crossed offsprings. So if was no crossing over occurring, then we should have only seen this, the parental type offsprings. That goes along with Mendel's theory. But we also observe recombinant offsprings due to the crossing over that occurs. And from this we can calculate the recombination frequency, which is obviously addition of these two recombinants divided by total number, and this gives us a number that is close to 70%. What I want to impress upon you using this slide is that particular chromosomes present on the same particular genes present on the same chromosome can cross over during meiosis, and thus give rise to recombinant gametes. So here is where the concept of linked genes come into place. What linked genes are, what linked genes tell us is that particular genes might be very close to each other, and when they undergo meiosis and crossing over, it can happen that they cross over together or do not cross over at all. They are so close. So if one crosses over, then another will also cross over. And this is indicated using the recombination frequencies. And thus, genes, so for example, genes for yellow body and white eyes are very tightly linked and show a recombination frequency of only 1.3%. Whereas the genes for white body and miniature wing are only show a recombination of 37.5%, and thus this shows us that they are loosely linked. And recombination frequency can have a maximum value of 50%. Okay, so is this slide clear? What I wanted to did you get what I was trying to say? So linked genes are ones that are present on the same chromosome, and depending on the how tightly they are linked, during crossing over they will either cross over together or not cross over at all. If they are loosely linked, then there will be a lot of recombinations, and that is why. So 37.2% means that the portion of recombinants is increasing. What the percentage means is that the total number of recombinants is increasing. That is, the genes can cross over independently of each other, and very tightly linked genes will not cross over at all or cross over together. So this is what the entire thing is telling us. Is this okay? Yes. So the answer with dealing with a so complicated slide, the answer is very easy. The if the recombination frequency is 10%, then the two genes are located on the same chromosome. So this is entirely what linked genes tell us. They must be first on the same chromosome in order for linking to occur or not. So if there is a recombination frequency, it means that they are located on the same chromosome. Is that okay? Then you want to yes. So in C. elegans, 50% of the progeny produced by mating with males are always males, whereas the more than 99% of the progeny produced by hermaphrodites without mating are hermaphrodites. This is because the four options are: 50% of the sperm produced by the males lack X chromosome, 50% of the sperm produced by males contain two X chromosomes, males have a higher rate of non-disjunction during meiosis than hermaphrodites, 50% of oocytes produced by hermaphrodites lack the X chromosome. So anyone wants to try and answer this? Shriyan says C. Males have a higher rate of non-disjunction during meiosis than hermaphrodites. Anyone else wants to try? I guess not. So this has been extensively discussed in the lectures itself, that how sex determination occurs in C. elegans. Sex is determined essentially by the ratio of sex chromosomes to autosomes, or we can simplify it as the number of X chromosomes as in C. elegans. So XX will tell us that the C. elegans is a hermaphrodite. XO will tell us that the C. elegans is a male. XO actually occurs by a rare disjunction, non-disjunction involving X chromosome, in which one of the daughter cells or the one of the gametes takes both the X chromosomes and one of them is left without any X, without one X, and thus this is a male. And males can mate with hermaphrodites, and this cross progeny are usually 50% males and 50% hermaphrodites. So this can be easily described with the Punnett square that is drawn here. So male has a X chromosome and nothing, so a 0 or a O, and hermaphrodites has 2X. When we mate them, we have two progenies with XX. So these two are hermaphrodites, and two progenies with XO. So these two are males. So irrelevant of what we do, whenever a hermaphrodite mates with a male, it will create 50% progeny with male and 50% with 50% of the progeny will be hermaphrodites. Is that okay? So essentially, the answer is number one. So the and if there was mating between two hermaphrodites or mating, so the hermaphrodites can fertilize their own embryos, and that is why they can produce progeny by themselves. They do not need a mating partner. So in that case, there will the sperm will be XX and the oocytes will also be XX. So all the Punnett squares, the progeny will be XX. That is, every one of them will be hermaphrodite. And thus, this explains that 99% of the progeny is produced by hermaphrodites without any mating are hermaphrodites, whereas 50% of the progeny produced by mating with males are always males. The reason due to this, the reason behind this is that 50% of the sperm produced by males lack the X chromosome. Is it okay? Yes. Okay. So that was all we had for today. The references to the slides are obviously still Introduction to Developmental Biology by Scott Gilbert, the ninth edition. And this is the YouTube link where everything is being uploaded. Anyone has any questions regarding the slides? I guess. So I would like to ask Jay, in the NPTEL portal itself, uh, there is a problem solving session link added where you can go and access it. Has anyone tried accessing the link? So in that there is a Google Sheet where the slides themselves as well as the video recordings are there. Kavya says yes. So you can just go ahead and type in and go ahead to the NPTEL site, and in the option there is a problem solving session given where by using which you can access the Google slides as well as the video recordings, or you can directly go to YouTube and search Introduction to Developmental Biology, and you will be able to look into the slides and also the videos. So if anyone does not have any other questions, okay. Anyone else does not have any other question, then we can end this meeting right here. Thank you everyone for joining. See you next week.